Um, Today we have this session chaired by Professor Steffen Borrmann, who's also based here at the University of Tübingen. Today he's joining us from the Center of Medical Research in Lambaréné, in Gabon, where he's leading a CHMI study on malaria. And yeah, with no further ado, I will hand it over to you, Steffen, and thank you for hosting today, for chairing today, and I hope that you all enjoy this session.
Thanks, Andrea, for the nice introduction. Yes, so without further ado, a few introductory words for Peter. Peter's the Deputy Director at the West African Center for Cell Biology and Infectious Pathogens in Accra, in Ghana. And you have a keen interest in SARS-CoV-2, the epidemiology, as well as different aspects of these infections, including immunology, I guess, Peter. And today, Peter, my first is in time. We have 20 minutes. Unfortunately, that's a 1 hour thing. And today, Peter, you're going to have a presentation for us on HIV 1 and 2 dual infections and what the implications of these dual infections are for treatment and potential cure of HIV. Looking forward to your presentation.
Thank you, Steffen. So let me just...
We saw your slides. I hope you can...
I'm sharing now.
Manage to project them.
I hope everyone can see my slides.
So, yes, we do. So thanks, Peter, again for volunteering.
All right. Sorry, Steffen, you keep breaking up.
The first presentation for today, the floor is yours.
Okay, thank you very much. Good afternoon, everybody. So before I go into my presentation, I'll just talk a little bit about where I work, which is the West Africa Center for Cell Biology of Infectious Pathogens. And it's a research and training center at the University of Ghana. And we use a consistent approach, which is molecular and cellular approaches to try and understand disease mechanisms. And we train PhD masters and postdoctoral young scientists. And the idea is to understand infectious diseases, human genetics, and non-communicable diseases. And to do this to apply it to diseases which are of interest to Ghana and Africa. And we're about 12 years old, and since inception in 2014, we've supported master's and PhD programs and postdocs up to 536 fellowships for people from 22 African countries in this period of time. And to focus on what we are here to talk about, the human immunodeficiency virus, HIV, is a lentivirus in the family Retroviridae. And there's two main types of HIV that have been discovered to date. There's HIV1, HIV2, and both of these are broken up into groups, different groups. In HIV, they're numbered based on the propensity or their contribution to the major, to the pandemic. Group M stands for group major, group N is not M, not O. Group O is an outlier group, and group P is a putative group. So most of the global pandemic of HIV is caused by group major, and which is further divided into subtypes of clades. And just to give a brief primer on the replication cycle of HIV. HIV is a pathogen of the immune system. So you have HIV GP120 recognizing the CD4 protein on CD4 T cells, and upon attachment, you have a conformational change which leads to engagement of co-receptor molecules, usually CCR5. Once you have engagement of CCR5, you have fusion of the viral envelope with the host envelope, and then you have translocation of the capsid into the nucleus. During the process of translocation, you have reverse transcription being initiated. That is the conversion of the viral RNA into proviral cDNA, which can, upon uncoating in the nucleus, can be integrated into the host genome. Once you have integration or insertion of the viral RNA into the host genome, you have what we call productive infection. And then basically viral mRNA can be made and translocated. You can have synthesis of viral proteins, packaging and budding. Sorry, let me try and minimize this. In a second, let me. Sorry, I lost my screen.
Sorry, Peter. We still
Okay. Yes,
see
yes.
the same
It's
slide.
fine.
Okay, good.
I was just trying to minimize something and I minimized the screen. Okay. So I mentioned that HIV primarily attacks the immune system, and if it's untreated, this is a curve basically showing the progress of HIV infection. Initially, upon infection, within the first 3 weeks, it's very difficult for a test to detect this because the viral load is extremely low, and the immune system has not yet seen this pathogen, so it has not yet had time to mount a response. So you have rapid viral replication happening, which is shown by this red line. And this is what we call acute infection. And at the same time, because it's attacking the CD4 T cells, you have rapid loss of CD4 T cells over the first few weeks of infection. And at some point, the immune system begins to catch up. You have a partial recovery of the CD4 cells, and you also have slowing of the rapid slowing of the viral replication. But without treatment, eventually the immune system becomes exhausted, and then gradually you have crashing of the CD4 cells and uncontrolled viremia and eventually death, which is the state where we call AIDS. You have high levels of opportunistic infections due to immune dysregulation. All right. Now, the two types of HIV have quite a decent amount of sequence identity, about 30 to 55%. But importantly, they differ in portions of the genome which is important, which the immune system sees, such as envelope. Envelope has a sequence identity sometimes lower than 35%. And then in terms of proteins that are produced, the main difference is HIV1 has viral protein U, VPU, and HIV2 has VPX, and we can talk a little bit more about the functions of these later. This is just a schematic of the virus. How I got involved in this is that my PhD work was looking at the clinical impact of dolutegravir, which is an integrase inhibitor. And I was actually one of the first people to work on dolutegravir outside the company. And that was in the lab of the late, great Mark Weinberg. And this is just a picture showing Françoise Barry-Sunusi on one of her visits to the lab. And the outcome of that work was to identify the R263K and G118R mutations, which have since shown up in the clinic as the main resistance pathways to dolutegravir. But during this work, we also noticed that the genetic background of the virus influenced the level of resistance and whether you develop R263K or G118R. And then knowing that West Africa has unique HIV epidemics, I thought that as dolutegravir gets rolled out, it's very important to study the West African epidemic, particularly in the context of dolutegravir treatment. I'll just reverse back and talk about why the difference between why you have high-level HIV diversity and the difference between the two types. So you can imagine that, you can imagine that in the context where you have, sorry, can I, 1 second, I need to tell somebody to pause for a bit. And just like 15 minutes. You want to make noise? Yeah. Yeah, we don't know noise, we just how many days. Okay, thank you. Sorry. So basically, HIV there's three things that cause diversity. You have an error-prone reverse transcriptase, you have high-level recombination, and what happens is that during recombination, during basically replication of the virus or during the formation of the cDNA, the reverse transcriptase enzyme tends to switch back and forth between different templates, and what you end up with is a chimeric cDNA. You factor in error-prone host RNA polymerase, and this is one of the reasons why within an infected individual you can have multiple quasi-species. But you can imagine that if you have multiple viruses infecting the same individual or the same cells during this process of cDNA synthesis, then you would have the chance to form unique recombinant forms. And once these unique recombinant forms are able to be transmitted from one individual to the other, then it becomes a circulating recombinant form. And West Africa is unique in that our major virus type is circulating recombinant form 02AG. And this schematic just shows how different viruses, different forms of CRF02AG are related to each other. And it's also colored to show the reported level of HIV-2 incidence. You can see that Ghana and Togo, where most of our sampling is currently done, have very related circulating recombinant forms, but also low HIV-2 prevalence. What's interesting is that looking at it over the years, as CRF02AG share of the epidemic has grown, HIV-2 prevalence has also gone down. And that's interesting, but another thing that's interesting is historically, looking at patient cohorts, it was noticed that in a dual infected individuals tend to live longer, or they tend to have a lag phase to AIDS compared to individuals who are singly infected with HIV1 only. And then that led to a hypothesis that maybe dual infection can have a level of protection against severe disease. And while it's not the focus of this work at present, also HIV-2 infection has an even longer asymptomatic phase, and much in some cases very rare to progress to AIDS. So we sought to understand how HIV-1 and 2 interact to reduce disease progression, thinking that could provide therapeutically useful insights. A lot of this work has been done by PhD student Edwin Montgomery. And the first thing was we wanted to look at how whether HIV-2 directly impacts HIV-1 infection. And some of this particular work had been done before, but the evidence was quite limited. So using the NIH AIDS reagent program, we did some cell lines, and we ordered different HIV viruses, subtype A, B, C, AG, and A1. And basically, let me just load everything up here. Basically, we used crystal violet endpoint staining to determine the TCID50 of the viruses, and then use those for infections with an MOI of 0.1 in luciferase and P24 ELISAs. And the question we wanted to see was, is it possible to have dual infections of HIV-1 and 2 in the same cells when you know that the virus that you have used the same co-receptors? And what we saw is that, yes, we can have dual infection in the same cells. However, we basically staged the infection, whether you have HIV-1 infection first or HIV-2 infection second, or simultaneous. And I'm not showing the HIV-1 first here, but if you look at subtype A, for instance, you can get significant simultaneous dual infection both for A, B, and C. But for CRF02AG, there's no impact of the HIV-2 on the levels of HIV-1 infectivity. Now, when we do a sequential dual infection, what we notice is that the level of reduction of the HIV-1 specific infectivity goes down for all viruses, and for CRF02AG, it actually becomes significant. And of course, this is still in an in silico, in vitro system. And we monitored in the sequential, we monitored to make sure that HIV-2 RNA was actually present in the cells. And this is just showing the reduction in infectivity for HIV-1 in the presence of HIV-2. And then, so that basically told us that there was a modest level of reduction. And it also told us that what likely happens in the patient is that you have HIV-2 infection, which is there before you have HIV-1 infection coming in. And we think that's important in the model that we're proposing. So then the question was, what stage of replication? And to do this, we can look at singly spliced or multiply spliced HIV RNA, and that will tell us whether and if the levels of these are changing in co-infected versus singly infected individuals. Then it can give us a hint to what stage that is being inhibited. So we basically
Peter, sorry, we can, if you please can already wrap up. So 1 more minute to go. Sorry.
Okay, sorry, Steffen, I got distracted by these people working outside. All right. So the long and short of it was that we saw that basically looking at unspliced HIV-1 RNA in dual infections. In dual infections, you had a significant reduction in the levels of unspliced RNA in the dual infection. But the multiply spliced, it's the story is a bit complicated, because CRF02AG is the virus that is relevant in West Africa, but it seems to have the least impact on the levels of inhibition, right? And HIV in subtype B is actually the opposite impact. But when we analyze it, we think that what's happening is that you may have HIV-2 accessory proteins that may be interfering with the action of HIV-1 accessory proteins. Because in the early stage, you have TAT, REV, and NEV, which are REV-independent proteins. And if they're produced, they tend to be produced from the multiply spliced viral RNA. And especially for HIV-1 subtype C, you can see that there's a huge reduction in the production of these. So it gave us a bit of a dirty answer that HIV-2 is directly impacting HIV-1 infection, but we don't know whether it's because also we don't know whether the same cells are infected by the same virus or whether they are just subsets of differentially infected viruses, right? So at least one thing that we could take away from this was that the co-infection led to low viremia. And we wanted to study this in patients, and basically we built up a long-term infection cohort in Ghana. And this was led by another PhD student, Mark Apieni. We've currently characterized the cohort very well. But when we wanted to pull up HIV-2 infection from it, what we noticed right away was that what was the clinical diagnosis? Obviously, we always confirm in the lab when we collect the samples. The clinical diagnosis of HIV-2 was different from the confirmed diagnosis. And long story short, what we found is that almost every sample we had, which we had identified as HIV-2, was either dual infected or HIV-2 was undetectable, and we're detecting HIV-1. So Helen Kamasa, another student in the lab, mentioned that Togo has a lot of HIV-2, and we ended up doing a lot of recruitment from Togo. And we also had to optimize our testing regime using different test kits and also developing an in-house PCR. I'm not presenting this, but an in-house PCR to allow us to properly define, allowing us to properly define the samples. after doing all of that work, we had 19 pure HIV.
Peter, I'm going to be polite. Sorry, Peter. Another 2 minutes, but then we have
Yes,
no
yes. So let
questions.
me just jump
Yeah, 2
through
minutes,
to
but no questions then.
yeah. Okay. So basically, we wanted to see whether there's an impact of the immune system, on the co-infection in these individuals. And what we saw is that the dual infections have a distinct cytokine profile from the co-infections. Of course, as expected, you had high level IP10 for all of them, but beta chemokines were more elevated in HIV-2 and dual infections. And we think that they may be competing for binding of HIV with the CCR5 receptors. And that may also be one reason for low pathogenicity. Okay, I'll skip this one. And then we saw that there were distinct profiles for IL-17, for example, which we had already seen when we profiled the cohort. And in HIV-1, it didn't really correlate in this case. We did flow cytometry, and there was evidence of lower CD8 T cell activation in the HIV-2 and the HIV dual infections, but unfortunately, we didn't do PD-1 staining, so we don't know whether this activation is coupled with exhaustion. But we also saw that there was lower levels of HIV-stimulated cytokine production in both the dual and the HIV-2 individuals. But what was quite interesting here, I don't know if I'm highlighting it, is that in the dual individuals, when we look at levels of CD19 B cells following stimulation with HIV-1 or HIV-2 and peptides, the main thing that I found interesting was that we see a slight increase in the B cell proportions. And so far as I know, there's not been any work done on B cells in HIV-1, HIV-2 dual infection. So we still have almost the same questions that we started with. We have some answers, and we think there's a B cell role to play in the HIV-2 impact on HIV-1 infection, which hasn't been studied before. And let me go to what we are trying to do now is drill down to see what steps of HIV-1 that HIV-2 is actually affecting, and then also look at the B cell regulated immune modulation, and this is by doing B cell sequencing and also looking at the neutralizing activity, producing monoclonals from the dual infected individuals and seeing whether they have cross-neutralizing activity. And this is because one of the hypotheses that has come out of our work is that we think the presence of cross-reactive antibodies may be why you don't have such a large pathology in the dual infected individuals. I'd like to thank the funders. There's a lot of people to thank on this slide, but I'll leave it here. And currently, what we're trying to do to augment the diagnosis is also we've done some sequencing of our cohorts. The HIV-2 has been very, very challenging to sequence, and I'm currently in Oxford to work with Dr. David Bonsall to develop a protocol to sequence that properly. Thank you very much for your time. And again, apologies for the disturbances.
Super, Peter. Thank you so much. Fascinating data. Unfortunately, since we're already over time, even without questions, we have no time to take questions. I also would have questions. And we have to switch immediately. Thanks again, Peter. Pathogens from viruses to parasites. And I'm very happy to announce our second speaker for today. Typically, we have a senior colleague and a junior colleague. Today, we have two senior colleagues. We have Jordache Ramjith there. If you, Andrea, do we have Jordache?
Jordache is here, but
Yeah,
you're
I'm here.
Hello?
I don't know. I can hear the connection very bad from your side, Steffen, so I don't know whether I should turn my video on or just leave it off.
Yeah, very
I'll pop it on so you guys can see my
maybe.
face.
Yeah, I think, Steffen, I think you can switch off your camera because it's your connection, the one that is
Yes.
having a bit of trouble.
Okay. Can you hear me?
Yeah, now it's better.
Okay. So my pleasure, Jordache Ramjith, to announce you. Thanks for accepting to give a presentation here to this round. You are Professor of Biostatistics at Radboud University in Nijmegen. You focus in pathogen host dynamics, and today you're going to present data from your modeling exercises in controlled human malaria infection studies, and we keenly forward to your present of this job to stay time, so 20 minutes questions. Thank you so much. The floor is yours.
Hi, thanks. Okay, so I'm actually from a slightly different background than I think most people that present here. So I'm a statistician. I have been working in infectious diseases for a while, but I'll try to keep the math a little bit at bay, and even when it's there, you don't have to be scared. So today I'm going to be presenting a Bayesian hierarchical model for analyzing parasite and gametocyte dynamics in controlled human malaria infection studies. And the aim is to estimate biologically interpretable quantities like parasite growth, hidden parasite onset, gametocyte commitment, maturation timing, decay, and the proportion that are male and the proportion that are female for the gametocytes. And we also want to compare these between malaria-naive and malaria-exposed study settings. So why study controlled human malaria infection dynamics? Well, the biological question is how asexual parasites dynamics translate into mature gametocytes and ultimately transmission potential. Field data are often very difficult for this because the infection time is usually unknown and sampling is really irregular. Prior exposure varies between individuals quite a lot, and CHMI gives us a much cleaner setting. So you've got the infection under control, you follow up very, very often, so quite densely, and we can observe both total parasite densities and sex-specific gametocyte densities. So the modeling goal is not only to estimate parasite multiplication, but also to connect parasite history to later gametocyte emergence. So we here at the Radboud UMC, the Radboud University Medical Center, also had a CHMI study. So I'm going to start with this. There were 12 malaria-naive adults infected by bites from Plasmodium falciparum infected mosquitoes who were followed from day 4 to day 51 after infection. We had really dense sampling around parasite growth and treatment, roughly half daily to quarter daily during the acute phase. Then we had much more spaced out follow-up visits later on. This data set is very useful for model development because the early parasite growth is fairly clear and the later gametocyte signal can be interpreted in relation to that parasite history. So we could really see that things are working really well in this perfect kind of scenario. So existing CHMI models are usually cycle-based approaches. A natural way to model parasite dynamics is to explicitly represent parasite waves or the multiplication cycles. We see waves because the young ring stage parasites, they circulate in the blood, but older parasites tend to hide away in small blood vessels, and then they burst and release a bunch more young parasites into the blood. So you've got the next generation that are much higher. So we see these successive parasite waves happening. This is biologically appealing, especially in the Radboudumc CHMI, because the waves are very visible. But there is a practical limitation because when you're using these kinds of models, you have to basically specify how many waves you visually see in each of those individuals. Yeah, so that becomes a problem really when the data might be a bit less clean, or when individuals have really different patterns, for example. And here we've got the Mali CHMI transmission study. So this is the second data set that comes from a study in Sotuba near Bamako in Mali. This is a peri-urban population with prior malaria exposure, so not like malaria-naive like the Radboudumc. And the study was designed to evaluate gametocyte development and transmission to mosquitoes. Here, 32 participants were followed from inoculation to around day 36, I think to about 49, with near daily sampling through the main parasite and gametocyte growth period, and later on, less frequent sampling. Compared with the Radboudumc mosquito inoculated cohort, here we see really that the trajectories are much more heterogeneous and less clearly do we see cycle structure happening. So those kind of 2-day waves that we expect, we see it a lot less clearly. So this is exactly the situation where a model that depends on choosing the number of cycles becomes difficult to use. Estimating parasite growth. So this really brings us to the question of parasite multiplication rate, or PMR. There was a paper that compared statistical models for PMR estimation, and they looked at basically using log linear models, which is the log parasite densities over time, versus sine wave models, which is a representative of modeling waves. And their key message is that the log linear and cycle-based models gave almost identical growth estimates, while of course they also found that fixing the intercept to inoculum size can bias the estimates, so it is better that every individual has their own intercept. Yeah, so for our purpose, we don't need to explicitly model every parasite cycle in order to estimate the growth. We could use a log linear growth model with individual level random effects, for example. Now, what did I do? Please don't get scared. Essentially, I wanted to jointly model everything in a Bayesian framework. So I wanted to estimate the parameters that we need, so like multiplication rate and gametocyte commitment, etc. But I wanted to do it, all jointly. One of the challenges I was having practically was that when you do things jointly, it also means that the gametocyte stage tends to feed back into the parasite stage, and it was causing some drifting in the original estimates that didn't look to be real. So I did a sequential Bayesian model, which is essentially saying, let me first model the parasite and timing stage, and then I will use those as prior anchors in the next stage when I model the gametocyte densities. It is still jointly modeled, so we're still taking into account all the variation from the first step into the second step. And how we do that exactly, you can see on the left-hand side of your screen, we have this latent parasite trajectory. All we're saying is we have some parasite load that we start with, and then they grow after some time TS. They start growing at a rate of R. And then we also say, yeah, at the start time has some distribution. The time from the start time to gametocytes time has some distribution, and we're kind of modeling both of these processes together. Yes. And then to model gametocytes, we say that first we're going to look at gametocyte emergence rate, which is a measure of the commitment times here, the derivative of the parasites is actually, it just means the amount of new parasites per point of time. And this little part here says that the amount of new parasites at a specific point in time appear sometime later as gametocytes. So committed parasites appear as gametocytes at some point later, and this is how much we expect there to be. And then we say that these gametocytes that appear can also decay after they appear, and they can decay at this rate minus k after some time of appearance. And we say that a constant proportion of these gametocytes are males, pi, and 1 minus pi would be female gametocytes. Yeah, and then we come to the interesting thing. The Radboudumc versus the Mali comparison. Here I showed that the main posterior estimates comparing the Radboudumc and Mali datasets. I focus on the broad pattern rather than every row, but what we could essentially see is that the Mali estimates suggest earlier detectable blood stage parasites, higher initial blood stage parasite density, lower parasite growth, thus PMR, the parasite multiplication rate is lower, shorter gametocyte sequestration time, and much higher gametocyte commitment. So these are not p-values in the corner. What you see here is just the proportion of posterior samples that are lower for the Radboudumc compared to Mali. And really, these results are telling us that these two data sets, or at least the malaria-naive cohort and the malaria-endemic CHMI, are giving us very, very different results. Here we can see a plot of the trajectories, Radboudumc and Mali. This is essentially to show you that the models fit really well. Of course, they're not perfect. There are some things that I would want to maybe look at a little further, and I'll talk about that in the last slide. Quickly, we also model the effect of covariates. I think that's something novel that we've added to these models that we haven't really seen before. I quickly added some covariate effects for the Radboudumc CHMI, and essentially we could see sex differences for the parasite growth rate. So male humans versus female humans, not gametocytes. But we could see some differences in their initial parasite load, in the parasite multiplication rate, and in the gametocyte sequestration time. We could see also that age had some effect on the initial parasite density load. Yeah, and next steps, I think one of the things I saw that was interesting was gametocyte commitment and the decay were kind of trading off with each other. So I'd like to look at a simpler no decay model versus a model with individual level decay. So that means essentially giving each person in this Mali dataset, for example, their own decay rates to see if that fits better and also explains a little bit more. Yeah, lastly, I've also built an app so that other people could be able to just load their data and choose their variables and be able to do this quite easily on their own. I think in order for me, I'm just sharing a screen now, but I could peep into that window quite quickly for a second. Just gonna share my screen. So this is what that app looks like. You can upload your data, choose covariates, etc. And I will just show you the results table where we could get lots of different things. Yeah, that's it for me. Thank you.
Many thanks, Jordache. Very clear presentation, but it was also very quick. So I have a question, but first the audience. But anyway, applause. Well done. Very interesting data. Looks very promising. Are there
Thank you.
questions? No.
Yeah, also sorry that I went a bit fast. I think
No.
12 minutes was
So this makes sense. Also, congratulations. Are there any? Let me see in the chat. Not quite. Maybe then I start. Can you please put up your slides again, Jordache, so that we have
Yeah.
something to look.
Yes, sure.
So my question
will
was,
share
I
my
wasn't really
screen.
sure whether I fully grasped this, but it looked as though, if I remember correctly, the slide where you had your model outputs and you were comparing time to parasite detections between Nijmegen and Mali,
Yeah.
Netherlands and Mali, and it looked as in the non-immunes it was shorter. No, longer. So exactly longer. This is
So
what
yeah,
I was very curious.
the malaria-naive cohort had later detection of parasites. Yeah.
The malaria-naive had later. Yes. Yeah. What is this? Do you have any quick explanation for this?
Yeah, I
This is
don't
not
know. I
unclear.
mean, I
Didn't expect
also
this to be.
Yeah, I also had some questions that I think I want to talk to some of the biologists with to make sure that everyone in the Mali was actually mosquito inoculated. Because of course, if some of them were blood injected with the parasites, then it could explain why they have faster appearance of parasites. But yeah, it wasn't what I would have expected that Mali would have had later. Um, showed parasites later.
Later, yes, later. I would think so. Sorry,
Yeah,
but
yeah.
if I do this research, this time until the blood cells parasites become detectable days, so it's longer in the non-immunes compared to the semi-immunes.
Yeah,
In
yeah, yeah,
your
yeah.
model
Which
also.
is not what I
So
expected
it,
either. But
yeah.
what I think so I have to talk with the colleagues that have worked on this data. But what I think could be happening is that maybe not all of the Mali are mosquito inoculated, so they didn't receive bites, but maybe some of them were injected because injecting parasites into the blood versus mosquito bites will lead to much earlier parasite detection times.
Yeah, sure. Anyhow, we can move on. Melissa, please, next, and then Bala. Melissa, please.
Yes, thanks, Steffen, and thank you very much for the presentation.
You have to unmute yourself, Melissa.
Can you hear me? I am unmuted. Can you hear me?
Yeah, we can hear
Yes,
you.
yes.
Okay, thank you. Yes, so thank you very much for the presentation. So I think there are caveats here, like so the Mali data, as far as I understand it, that was blood stage infection. So you would expect that they would have earlier detectable times, but just have a look. But I think this Mali CHMI data is actually the induced blood stage model. So
Yeah,
you
that's
would
what I thought.
expect earlier.
This was my questions when I got the results. I thought, yeah, when I did the final model, I thought, maybe this is blood stage. It's the only thing that could explain why they have earlier parasites. So
Yeah, it was a blood
thanks.
stage infection.
Okay, perfect. Thank you. Yeah, that gives us the answers then.
I'm sorry, I was offline for a short moment. Melissa, you are suggesting, or you know, that this is induced blood stage infection and not mosquito bite or sporozoite challenge?
No, it wasn't. It was blood stage infection. This is because I've noted you've indicated that it's Woodford et al., right?
Yeah,
Yes. So this was a collaboration with the team in Australia on blood stage. But
Yeah.
please double check. But I suspect
yeah,
this Mali
yeah,
study was a blood stage infection study.
yeah. I'll double check, but I think so too. I think so too. It's the more obvious explanation. But then if we had to compare it to the blood stage parasites at the Radboud, I have that data somewhere as well. I can quickly tell you if there'll be a difference. Just if you're curious. Uh, I've got my table somewhere here. Yeah, at the Radboud, I think the blood stage was about 4.7 days, which is a little bit earlier than if this is
Yeah.
Mali blood stage, then it's a little
So
bit
it's
earlier.
expected. Yeah, no. So I mean that makes sense because ideally you do see delay. You would expect a
Yep.
delay with the um, immune experienced individuals compared to the
Yeah,
naive.
yeah,
So I had a question more around your analysis and how you're calling your parasite multiplication rates. Because traditionally, I think, well, not traditionally, I mean, we kind of know that there is in immune experienced individuals, you don't really see the traditional dynamics of parasite growth.
yeah.
So how are you accounting for, and I know you've talked, oh yes, I know you talked about sort of making sure that you're not, your model doesn't include, or it takes into account that there will be points that are missing just because of the nature of the data that we get from these immune experienced individuals. So how is your modeling dealing with that?
Well, I think for the parasite multiplication rate, we calculate it from a growth rate, right? And that growth rate, we say, let's just instead of looking at waves like we would in those wave-based models, the log linear models just says, let's fit a line through all the parasite trajectories, and that should give us the parasite, well, the growth rate, which we transform into a 48-hour parasite multiplication rate. So my parasite multiplication rate is just a transformation of a growth rate that's calculated through a linear model on I hope you guys can still see my screen, but on the parasite trajectories. Of course, for the gametocyte, the Mali data was a little bit different because gametocytes had already started at some point, so they weren't purely asexual. And I had to really take into account in the measurement area part of the structure that after gametocytes appear, that these were no longer solely asexuals, that they were total parasites as well.
Yeah, but I think my question is largely linked to the rate that you're calculating.
Yeah, yeah,
Because yes, the rate will be calculated based off over a 48-hour cycle.
yeah,
But there will be instances where you will have missing data, well, not necessarily missing data points, but you won't have data points just because of the nature of how parasites grow or how we're able to detect these parasites. in these individuals. So how are you accounting for that?
But that usually will have just a smaller influence on the intercept. So if you can imagine, like, let's just look at one block of data from the Mali. Let's look at just this one. If we had to say that parasites started at this point, and here these might be, let's say these are all the peaks of the parasite waves that we're not seeing the rest of it, the going down parts. It's still fine to have a straight line go through the peaks because that's just the rate of growth rather than affecting the intercept. So here, the rate of growth wouldn't really matter that some parasites are sequestering and we don't see them.
So I guess my other point was to ask whether you have sort of within your analysis some rules about X number of time points required to be able to see detectable parasitemia.
Yeah, I think this is a difficult thing because the whole thing that we've done is built around CHMI, which really usually has regular sampling. I wouldn't.
I understand in the naive population, you will see fantastic parasitemia at every time point. And a case in point is actually the data you're showing us here, where you see zero parasitemia after somebody has had some detectable parasitemia.
Yeah. I mean, but those are treated as being below the level of detection. So I treat them as left censored, not as being invisible. They still contribute to the likelihood that gets our parameters estimated, but they are counted as being below the level of detection. So I treat them as being left censored rather than being negative.
Okay.
So mathematically, it's just it goes into the likelihood as a measurement that's undetectable rather than a measure that's not there at all.
Mhm. Many thanks, Melissa, also for your questions. I guess
Yeah, thanks a lot.
we have hit the 1 hour, 60 minutes. Thanks for joining today and hope to see you soon again. Thanks again, of course, to Peter and Jordache for presenting today, and to everyone. Have a nice remaining day and see you soon here. Thank you. Bye-bye.
Thank you, Steffen.
Thanks. Bye.
Thank you, everyone. Bye.